Okay. All right. Hi, everyone. Thanks for joining us again for another week of seminar. So this week, Dr. Gretchen Gerrish is going to be giving us a talk. Gretchen is an evolutionary ecologist who became the director of UW-Madison's Trout Lake Station in 2019. Her research focuses on how aquatic organisms adapt to change in ecological conditions. Her work involves reconstructing land use change and food web changes over time. She also studies how solar and lunar light affects feeding and reproductive behavior in zooplankton. She earned her master's from University of Illinois Champaign in natural resources and environmental sciences, and a doctorate in ecology and evolutionary biology from Cornell University. Before joining Trout Lake Station, she was a tenured professor at UW-La Crosse. And with that, I'll kind of lead it over for you. And I don't know if you're going to explain a little bit too on how Trout Lake is associated with the CFL, kind of or not, but whatever works. So with that, I'll let you take over.
All right. Thanks, Olivia. And thanks for the invite to participate with your group today. I'm really excited to share some of the work that we've got going on at Trout Lake. It was also fun to connect with Olivia, who's a Trout Lake Station alum. So super fun to see her in her next steps. As Olivia said, I joined the Center for Limnology, which is at UW-Madison in 2019. And I'm located at our Trout Lake Station field site up in northern Wisconsin in Boulder Junction. We're a year-round field station that operates mostly on lake research, although we do have plenty of terrestrial wildlife and natural resource partners that use the station for different types of projects as well. When I got up to here, coming from La Crosse, Wisconsin, I was already aware of winter. But I think one of the things that struck me is how important lakes and ice and snow really can be. This year is very much an indicator of how much snow can impact the existence of the lakes and existence of the humans interacting with those lakes. And so we developed a partnership. I've been working with Hillary Dugan, Noah Latta, Greg Sass, and Zach Feiner, who are all lead researchers within the CFL network and the Wisconsin DNR group, and Ellie Socha, Adriana Gorski, and Emily Whitaker, graduate students on the project, to ask about phenological whiplash in lakes and how lakes are responding to highly variable ice and snow conditions. So it's probably not a lot of news to folks who are viewing from Minnesota or the Twin Cities that snow and ice have been highly variable in recent years, this year being no exception. And ice is a huge part of our existence in the North Woods on the far left here, from early fall when people run out on that earliest ice to get the shiniest, fastest skating, to both recreational and subsistence fishing throughout the winter, and access to the food resources underneath the ice is a huge part. You'll find entire villages of folks camped out on the ice for the duration of winter. And recreation. We are very dependent on snow and ice conditions throughout the year as humans, and the same goes for our non-human residents in these systems. And many of us aren't willing to give up the ice and the snow and the winter. This picture was from a month ago when we had the very warm temperatures and thaw. And it's from Lake Eau Claire, and it's a social media post, but there's a person out here on the ice with their truck, and it's all open water below. And so when we think about variability, again, no one expects at the beginning of February you wouldn't be able to go out and ice fish or have access to those systems that you have for a very long time. But as many of you probably are aware, we are losing ice, and we're losing ice duration in our winters. And so the story that I'll talk about today really has to do with ice dynamics and the change of snow and ice dynamics over both long and short time scales. And so the trend of losing ice over long time scales is one that's been well supported in the literature. This figures from Feiner et al., but a lot of work here at Trout Lake Station done by John Magnuson since the 1960s, and ice records going back over 100 years with the Wisconsin DNR and observers down in on Lake Mendota, have recorded the duration of ice and the timing of ice off over the years. And this figure shows the ice off date over years, and you can see that there's a decline. And based on the references that we have here in the Midwest, the decline is about a day per decade. And so over the last 100 years, we're looking at least a 10-day change in the duration of ice on our lakes. And this pattern is not unique to the Midwest. So here is a broader look at thaw date, so that ice off date. And this graph is oriented in the opposite direction. But here's March 1st. And what you see is that all of these lines are trending upward. We're getting earlier and earlier in our average ice off date. And if you look across many of these systems, you also see that the trend is about a day per decade that we're losing in terms of our ice duration in the spring. So while we're losing this, while this long-term climate trend is readily available, and this record is actually from the EPA. This is one of the indicators of climate change that's used in the global discussion about climate change broadly. What we also see, and what struck us, is that we've seen in the recent years a 4 to 5 times increase in the variability in ice off date. And so here are data from Lake Mendota, Minnetonka that are longer term, and then Sparkling Lake and Mille Lacs. And what you can see is this is the rolling variance in ice off date. And you can see that the variance was somewhat consistent. And then within the last 10 to 15 years, we've had almost a 5 times increase in the year-to-year variability in when ice goes out. This was evident in the last couple of years. So just last year, we went out in late April up in this region. And in 2021, the ice went out in mid-March. So we had almost a 50 or 30-day difference between 2021 and 2022. Variability in ice off can do a lot of things to a system. And so a lot of what we'll talk about today has to do with this variability, both in ice off, what's causing that, and also the implications for the ecosystem and food web. So just a little bit of where we are in location. So I think viewing from down in your area, we're a short 3.5-hour drive up through the center of the state, nestled tightly within the ceded territories, about 10 miles distance from the Lac de Flambeau Reservation, and on the south shores of Trout Lake. So here's Wisconsin, here's Trout Lake. The station is located right here on the south end of this lake. And then we have 3 lakes that I'll include in discussion today. Escanaba Lake, which is up here to the right. This site is where the National Highlands Fisheries Research Unit is for the Wisconsin DNR. And they have long-term data on many aspects of the lake, including ice off, fish spawning, zooplankton, phytoplankton from Escanaba Lake. And then Sparkling Lake and Trout Lake are 2 lakes that are part of the North Temperate Lakes Long-Term Ecological Research Program. And they have continuous data on, again, all the water chemistry, physical components, zooplankton, phytoplankton, and fishes since 1981. Those data sets will be focused on here. And then I'll talk a little bit about this project, which is literally in process in terms of data collection. So to get us started, I'm going to talk a little bit about ice and snow-covered seasons and thinking about them as actually important periods for lakes. For a long time, it was thought that underneath the ice, things would go dormant or slow down, and that some of the food web dynamics that we understand would almost rest for winter. There were a lot of reasons for that. One is safe accessibility to the systems. 2, the harsh conditions that exist for people going out to research and sample, and just the kind of the unseeability of what's underneath. Over the years, we've begun to build greater and greater under ice understanding, and anyone who ice fishes readily recognizes that things are actively processing underneath the ice. As we explore the under ice dynamics, some of the questions that the group's considering is how does fall ice formation and the subsequent snowfall influence our ice conditions and that subsequent timeout or timing of spring ice out? And second, how does ice thickness, clarity of ice, and snow cover dictate what's happening below the ice and set the stage for spring, both within the physical and biological communities? Based on the LTR data records, ice formation, this is a image of ice and snow formation over the history of the LTR. This is from Sparkling Lake, which is one that I'll feature today quite frequently. And for folks who may not be as familiar with lake and ice dynamics, I think it is interesting and something I've become almost too acquainted with living up here, is that the ice formation, so we break ice down into a couple ways. When you think of ice formation in the fall, you think of the water surface, so hopefully you can see my cursor moving over on the ice here. This level right here, if you look below this level of ice, this is a chunk of ice that was cut out of the lake this winter. And if you look at the ice below here is what we would call blue ice or clear ice. And that ice actually, this would be where the water, the surface of the water would exist. And this ice grows downward into the lake. That's the original formation of ice in a given season, is you'll have the lake surface, it freezes over and then freezes down into the system. And so this clear ice allows for much more light transmission. And it also is a very strong structural ice that will hold up pretty strongly in the lakes for a long period of time. In regions where we have a lot of snow, we get on top of this white ice or blue ice. I think I'm referring to a blue ice throughout here, is where we get white ice. And so the white ice is basically snow that then falls on top of the blue ice, and then it packs in and solidifies as air temperatures kind of thaw and freeze. It forms this white ice. And light has a harder time getting through the white ice. It's not as clear, and it can be quite a huge layer over the course of the winter. And so just to kind of set the stage, I wanted to show that the impact of blue ice and snow can actually drive the structure of the ice, both physically, and then that can then subsequently drive what's going on underneath the ice. So here we see that in some years, this is our January snow and ice steps. In some years, when you have low snow or lower snow, you can actually get a lot of formation of that blue ice early in the season. That means you've got a really strong ice layer, a little bit of snow on top, and that would be stronger and last longer through the season than in years where you maybe have a little bit of ice and a lot of snow sitting on top. So the interaction of ice and snow is one that we're just beginning to really understand how that then impacts things underneath the ice. And one of the studies that was undertaken in the last 3 years here near Trout Lake Station with scientists from the Center for Limnology was led by Hillary Dugan, Adriana Gorski, and Ellie Socha. We're graduate students on that, and they worked to clear an entire lake of the snow every time it snowed all throughout the winter, and ask how does that change the ice formation and the conditions underneath the ice. And so here's a picture of them using an Argo and a snowblower to shovel and plow the entire surface of South Sparkling Bog, which is nearby. Here's Ellie with her zooplankton net. And so we are going to talk a little bit about those results. The first year was considered a reference year, and so the snow was allowed to pack in on the lake. So here's a picture of our reference year. And as you can see, we had some good black ice in that year, and the black ice grew. So here's January, and then that black ice, or the blue ice extends down into it. We're calling it black ice here. Extends down into the lake. But then it snows, and you can see that the snow starts packing in as white ice on top of that black ice. And so by the end of winter, you have a little bit of black ice, a ton of white ice, and a good pile of snow on top. In the first year of manipulation, they plowed and plowed, but in the first year of the manipulation of 2020, when they started plowing, we already had pretty good, the ice started forming and it snowed right away. They got out there and plowed as soon as it was safe, but there was already a pretty good layer of white ice that had packed in on top of the black ice by the time it was safe to get out there. And so the black ice maintained, white ice was still present, and the snow removal did continue, but we still had a good amount of white ice in relationship to the black ice that was there. But by the end of the season, we had pretty much transitioned to all black ice as it started to thaw. In 2021, we had a stronger black ice level in the beginning of the winter, so we had a lot more black ice accumulate before any white ice could set on top. And so we had a larger block of black ice, a little bit of the white ice on top, and again, tried to keep the snow off throughout the year. And the implications of this were seen. So the early season ice characteristics in the snow year, you can see we've got three things here. PAR is our photosynthetically active radiation. And in our first year, our control year and our manipulation year, the first one, that white ice really didn't allow a ton of light through. In the third year, where we had more black ice, we see that the photosynthetically active radiation reached a lot deeper into the water column of the bog. In relation to that, we also see that the chlorophyll in both of the manipulation years, even just removing that snow off the top, even with some of that white ice, we still see an increase in the phytoplankton or the chlorophyll A content. And again, in the black ice year 2 manipulation, we see more chlorophyll. Ellie's project really focused on why things were changing and how it changed specific to the organisms that live there. And so here are some of her findings in the plankton data. So again, as we go through, here is the control year where the snow was allowed to accumulate. Here's the white ice year, and here's the black ice year. And what you can see is we had substantial more algal growth in the black ice year, and that was driven mostly by chlorophytes, or the green algae. These are usually favorites of the zooplankton to eat and require high light conditions or higher light conditions to thrive. But then we also had some cyanophyte bloom, and so the composition of species was dominated by those two groups, in contrast to our control year, which had a lot more chrysophytes. In addition to the phytoplankton composition changing, Ellie counted zooplankton throughout the manipulation period. And you can see that in the control year and the white ice year, the levels of plankton were very low. And so there are tiny bars there, but the compositions were very, very scattered, and the abundances were extremely low in terms of densities per liter. In the black ice year, with all of that phytoplankton and light coming through, we saw a dominance of two species in particular, Ascomorpha and Asplankna. Those two species, so here in the light green and then in the dark green, as well as some Branchionis increases, these species really thrived in that last year. So we saw a pretty large transition in species dominance. The two dominant ascomorphs and Asplankna weren't even hardly detectable in the prior years. And so Asplankna is known to be a predator on ciliates and other rotifers. And what we think is that that phytoplankton bloom allowed for different species of rotifers to then come in and outcompete, as well as stimulated the microbial loop, which was not measured in this study, but would be a way to explore this finding further, is how the microbial loop then probably played back into the zooplankton species composition change. So really the take-homes are outlined in the figure that we have here. So when the snow covers with, we get more white than black ice. And so when we have no snow or low white ice, we get thicker black ice, higher light penetration into the water column, and that higher light allows for chlorophyll A to increase and higher dissolved oxygen concentrations. The phytoplankton community increased in diversity, total biovolume, And most of that was driven by the chlorophytes and cyanophytes that were reached. And the zooplankton community changed also in terms of its species composition, and its density went increased by almost 500 times in terms of biomass. But the diversity actually went down. It allowed for kind of monostands of this Ascomorpha and Asplankna taxa. And so you don't see as many different types of rotifers, but you get a couple that are really dominating in the clearer years. So our take home is that the under ice environments and species-specific responses are highly dependent on ice formation and snow dynamics. As we think about kind of the seasonality of lakes, with that in mind, this is what's setting the stage then for our spring ice-off and that high variability in our ice-off date. And so I know that this is kind of a mixed audience of terrestrial and aquatic folks. And so just as a reminder of how things change. So we just talked about the winter period where we have ice on top and the water's pretty cold underneath. This is the structure of a lake. In the spring, what happens is as that ice melts, the cold water mixes down into the water column and actually stirs up the lake, gives it a little turnover here in the spring. It's a mixing period. That mixing period will stay as long as the surface waters are cool, but as the sun warms the surface waters, that's my point, as the sun warms the surface waters and we move into summer, the solar radiation at the surface warms the top layer and creates what's called stratification in the lake. And so you'll have this warm upper layer of water, the layer called the thermocline, and then the cold underneath layer. If you've ever swam in a lake, you've probably experienced the thermocline. That's why when you swim down to a certain point and all of a sudden the water gets really, really cold. And so this density differential can actually hold up throughout the entire system. And these are almost functioning as two separate water bodies where nutrient dynamics and mixing in these two layers maintain a pretty substantial separation in many of the lakes. As we move into fall, the temperatures at the surface cool, that cold water falls, and again goes through a period of mixes. This can be really important, stir up nutrients and allow for some fall algal blooms and fall spawning of certain fishes before it gets too cold and the surface begins to freeze. And so this is what we call lake phenology, so thinking about the transition of the lake throughout the seasons. But during that same period, we also have phenology in its more traditional use, where species and taxa change in their life stage as well as their population dynamics. And so here on the right, I'm not going to go into detail on all of these. The idea is that we see dynamics where under ice, this species is dominating. And then as we transition into the spring period, we have a few species that kind of take over. And then as we transition into our summer period, another group of plankton that take over. And many times lakes have these kind of transitional steps that are considered to be part of the system's history and food web dynamics. So ice-off is considered this critical transition period. The light regimes shift, all of a sudden light's available, the energy that's coming into the system, the oxygen mixing into the surface waters. You also will have this mixing where nutrients come up from the bottom. And of course, gas exchange is more readily happening. And so everything from the physics, the chemistry, and the biology of the lakes will change. I kind of mentioned this earlier, but just to make sure we're clear, phenology, especially if you're used to working on plants in a forest and or insects, oftentimes that term is directly applicable to the biological transition of species. In this presentation and in the literature, we also use phenology to talk about the transition of the lake conditions or ice phenology in response to seasonal shifts. So I'll be mostly focused on the second use of that term. Phenological whiplash is a phrase that we've put forward, and the whiplash part of it is still, I always like to debate terms. I think the reason that we use it is because it's being applied to a lot of climate variability. We've got the term weather whiplash being discussed, where you go from one extreme to the next. Thermal whiplash, mixing whiplash, you see it popping up, and so we've been playing with applying it to this lake dynamic. And that's because we want to contrast what's happening in these lakes in this year-to-year variability with kind of this longer term of ice, or this longer trend of ice off. And so here, that's what we're planning to share. I'm going to show a few sets of data. Most of it is in reference to the North Temperate Lakes Long-Term Ecological Research Program. And we're going to focus in on 2012 and 2013 as a case study, but then we'll also expand out and look at the data set over the last 40 years. But 2012 and 2013 were these iconic years of whiplash, and they were early, about a decade ago. And 2012, the ice went out around March 20th. And here's what happened at Trout Lake Station. It was before my time, so I won't own it fully, but I think there's folks around that can. And so they were headed out to Aliquash Lake, one of our common study lakes, never expecting that early in the season it would not be accessible. They did manage to get out and everybody was safe, but that was March 20th of 2012. And then in the very next year, the students began to show up at station. And so here's the tweet on the Trout Lake Station page. And so students are arriving in 2 weeks, and we get everybody out to do limno launch and field work that very first week, get them out, teach them how to drive boats and back trailers. And this is what the lake looked like. So that 50-day difference, if you can imagine that from a point of view of a phytoplankton, which maybe reproduces every day or every 2 days, and the amount of biomass that accumulates in 50 days, or the amount of transition in that population that could occur, or a zooplankton who maybe lives a week, thinking about how many generations zooplankton could go through in a 50-day period. It's really a huge change in thinking about this from 1 year to the next. And so I'm going to show some different data to talk about this 2012 and 2013 change. And so these data are from Sparkling Lake. And here we have 2012, which is when we had the ice go out early. And in 2012, you can see the ice off date. And then this graph shows the lake depth. So this is 0 to 15 meters. And for the period after ice off, what was happening is there was a lot of mixing that was going on. The surface waters would warm, but not enough to stratify the lake. And so those warm waters were mixing all the way throughout, and even the bottom of the lake was warming. By the time stratification set in, the water at the bottom was already, getting up there to 10 or so degrees. But then the surface waters warmed up. We had strong stratification. But this layer of really warm water set into 12 to 13 meters by the end of summer. If we contrast that with 2013, when the ice went out in May, what you see is there wasn't this early period of mixing. Instead, we went from ice to very strong warm temperatures at the surface, right? June started to warm up. And so stratification set quite quickly. And we got this density differential quite fast. The bottom layer of water stayed much, much colder in 2013. And the warm layer, that thermocline, only reached down to about 10 meters instead of 12 or 13. And while for us, it might not seem like a big deal to have a meter or so change or a 3 to 4 degree temperature shift, it's actually a huge component when we think about the energetics and the system itself. So here's, so that was 2012 and 2013, but as I said, I'm going to go between that as a case study and then some average information across the 40-year LTR data set. So here you can see that in red, we have our early ice off years, and water temperatures in those years are higher. This is the surface water, so they start out higher, but they kind of reach a plateau similar in the surface water. That deep water layer, where our early ice off often reaches much warmer temperatures in the bottom layers of the lakes. And late ice out years, here represented in blue, the late ice out years are always colder, and they remain colder in the bottom layers of the lake. And so we do see that extreme early and late ice off events can have pretty substantial differences throughout the season. Here's just another type of a way to present this. The later the ice off date here, the we get that mixing depth. It's going down deeper and deeper when we think about how many days we have for mixing down into the water. So another way to think about this is just energy storage, right? Temperature is energy. Things like phytoplankton and zooplankton usually grow faster in warmer temperatures. And fishes, everything responds to temperature cues in their environment. And so when we look at this as just internal energy or joules per meter squared, in 2012, we see here in red, we had a substantially more energy stored within the lake system for most of the year until we got into that fall winter condition. And so the other thing is we had higher levels of oxygen that were available to all the biota in that early spring. And so we have a shorter anoxic period. So again, a lot more time for primary productivity and a lot more energy to help ramp up those metabolic processes going on within the lake in the early ice-off year. Temperature, which we just discussed, interact with light and photoperiod, and all of these conditions are very biologically relevant. Oftentimes, when we think of biochemical processes, the rate of the metabolism, developmental rates, foraging and reproduction are often cued by temperature, light, and photoperiod cues. And so when we are having a 50-day difference in ice-off, what it means is that your temperature, light, and photoperiod cues can actually become offset. Up here, photoperiod shifts a lot, right? We're in the northern portion of the US, and while I recognize that farther north these things change even more, here's what we're looking at. This is pictures of 5 p.m. in the winter and 10 p.m. in the summer. So we range from an 8.5 light dark to a 14.5 light dark in the summer in this region. And so photoperiod can be very shifted. Here's some examples of things that respond to photoperiod. So the flowering and seed set of plants can really shift with photoperiod. This is one of the little critters that I've studied for a long time, a little Daphnia peeking his head out of a dormant Ephipia. So they produce these long-lived dormant stages, and the hatching of those dormant stages can be a combination of cues, which usually are light and temperature and photoperiod dependent. The spawning of many of our shallow fishes, so many of the spring spawning fish like the bluegill and bass and walleye that we care about, especially in our recreational and subsistence fisheries, are spring spawning fishes, and the emergence of insects, both aquatic and terrestrial. So going back to that 2012-2013 example that we talked about and the offset of potential temperature and photoperiod, here is what the lake temperature profiles looked like on May 16th of 2012 and 2013. 2012, our early ice-off year, here you can see that the lake was already stratified and almost, 4 degrees warmer by the time we reached May 16th than in 2013, which was our late ice-off year. That's confusing. Your day length is the same here, but you're looking at a 4-degree temperature difference in your habitat. So the zooplankton that may rely on a combination of cues would be receiving very different temperature cues on the same photoperiod days. And we see that these taxa are responding differently in the 2 years. This is a figure from Sparkling Lake looking at 2012 and 2013 data. And what you see is primary productivity increases in that early season, has a stretch of high primary productivity, and then does match up with the other taxa. The zooplankton also, they come out later in the late ice-off year. And if you actually look between the same levels of zooplankton diversity, you see that there's about a 50-day lag before the 2013 year catches up to the 2012 year in terms of diversity and in terms of biomass for our plankton. Here's a representation and a look at this across multiple years, kind of like we did with the other data. And so here in the dark bars are our early ice-off years, in the blue is our late ice-off years, and then the gray are the average. And what you see is that, again, we've got phytoplankton abundance peaking earlier in the early ice-off, average, and then later in the late. And then rotifer densities, which are some of the most abundant zooplankton in our system, we see that they peak and have a really big peak early season in those early years, and then our average years, and that in late ice-off years, the rotifers really seem to struggle getting a large biomass abundance. Cladocerans don't show as strong an impact because they're a little scattered, but what we do see consistently is that they don't have big boom and busts in late ice-off years that are timely in that early spring period in Sparkling Lake. So the plankton are responding, the temperatures are responding, and one of the hypotheses that we're actively testing and have hypothesized is how these offset dynamics between early and late years may impact food webs. And this match-mismatch idea is based on Cushing, 1990, who talked about how if we shift the timing of events, we could actually mismatch, so eggs and larvae, and then the availability of prey. If eggs and larvae are cued to go early, it could be that the prey are not abundant enough for when they come out and need to feed. And so if we apply that to our aquatic food web, here's just an example. In an average year, which is what they've evolved to for, a long time without the variability, in an average year, you'd have the algae coming in, the zooplankton coming in, and then the fish transitioning to feeding on those plankton coming in the subsequent timing. What could happen is in an early ice-off year, your algae peaks and is highly abundant early. Your zooplankton, depending on that photoperiod cue, comes in later and doesn't have enough food resources, and so maybe your fish are limited in their growth. In contrast, in a late ice-off year, maybe all your algae's there and your plankton's there, but your fish were waiting on the right temperature, and so they come in later and again might miss out on food or other things. And so this idea of food web changes based on mistimed cues or mistimed population dynamics is one we've been exploring. And one place we've looked at this is in the perch and walleye data. And so on the left here, you can see perch information, so fish length we're using as a proxy for kind of the growth and size of a fish throughout a season, and then the density of those individuals. And for the perch, it does look like all that food in the early year, the peak in the phytoplankton and zooplankton growth, and overall more biomass in the system. In an early year, we have larger perch. In an average year, we see that it's in between, and then in a late ice-off year, we see some limited growth in the perch, although they're still doing, pretty okay. In the walleye, the story is not the same. In the walleye, what we see is that in an average year, the walleye fish lengths are larger and more distributed. In an early ice-off year, the walleye are slightly smaller in their peak and not as distributed in size. And in late years, again, even lower. And so the walleye are growing less in early ice-off or late ice-off years. They're not following the same pattern as the perch in terms of their response. This was reinforced. Zach Feiner went into the data from Escanaba Lake and was tracking the spawn date and the age 0 walleye abundance as a proxy of population success. And so here he's looking at years and spawn date, and very comparable to the ice data. What you see is that the spawning date of year is getting earlier and earlier, about a day per decade. Surprise, surprise. So that kind of matches up with what we've been seeing in the ice data. And also that within the last 20 years, this is standard deviation. We see a much higher here, at least a doubling in the variability of our spawning date for walleye from one year to the next. So we matched this up with ice data and we asked, okay, if we have early and late ice out, how does that then interact with our days from spawning. And so here in red, we have 20, or we have in the dark red, we have extreme early ice off, and in the blue, it would be late ice off, and then the zero, or kind of the gray bar, the gray circles, represent our average years. And so if you look at the age zero walleye abundance, you see a lot of gray dots extending up to that 40,000 mark. And so you see that there's higher walleye abundance in average years compared to our darkest red and darkest blue circles, which are the more deviation we have from average spawn date, the lower the age zero walleye abundance is, or the lower the success of the walleye population. And so this is telling us that walleye recruitment or walleye survival to the next year is lower in our extreme early or late ice-off events, and they do better in average years. So just to kind of give an overall summary of this last portion, our physical template, we have an increase in the frequency of these events, the back-to-back early late events. We see a significant change in the energy budgets for the lakes under these conditions. Whiplash years have big differences in early season primary productivity, and this can alter species diversity within our zooplankton communities and ultimately change fish growth and fish recruitment. So where are we headed? As I said, this is a work in process. That first study was based on existing long-term data, but we also have hypotheses about the evolutionary outcomes of phenological whiplash. So when you think of having a year where the ice goes out 30 days earlier than the next year or 50 days earlier than the next year, in that early ice out year, the animals that are going to hatch from the sediment bank and take residence in the water column for the zooplankton are going to be ones that maybe have an early cue for hatching, or if you're a fish, an early spawning date that you're adaptable to. If all of the early genotypes and phenotypes are succeeding in that year, then what happens in the very next year when you have a very late ice out and all of your genotype phenotypes are the ones from the early season the prior year? And so it's possible that we could have what's called selective sweeps, where your phenotypic frequencies are either pushed to driven either earlier or later. Or the other possibility is that we'll have selection for greater phenotypic plasticity within the system. And so one of the questions that we're asking is, do we see selection for early versus late phenotypes, a loss in overall genetic diversity, or an increase in phenotypic plasticity? Ecological outcomes of this phenological whiplash is looking at community composition changes. We need to increase our frequency of spring sampling. Every 2 weeks or once a month isn't quite enough, which is what we have access to through the LTR database right now. So we've been doing either 3 times a week or weekly sampling to try to get better tracking of our community composition changes and those food web interactions. And also looking a little bit more at food web dynamics and isotopes to think about whether ecological buffering is pushing our maximum, how far we can push those mismatch or match dynamics before we experience species loss or recruitment failure in some of our important taxa, especially things like those big walleye, bass fisheries that people depend on. And we do feel like there's implications for management. And are there ways to manage our local systems for whiplash itself? Diversity, if that's a goal of management, keeping that diversity, we've seen that it can change from one year to the next. Habitat complexity and connectivity, also from one season to the next, rather than focusing on just the summer season when we're really aware, how does that transition from ice, snowy ice and ice into spring matter for our understanding of these taxa? And do we put as much emphasis on stocking programs or management strategies in years where we already know the system is going to be extremely stressed? And so understanding our harvest implications in these early versus late ice off versus average years, and thinking about being able to reduce other stressors in those times where the lakes may already be at their max in terms of a climate threshold. So as a take home, I think our ice-off variability will reduce adaptability to long-term climate change. And so with that, I think I'll give acknowledgements both to the North Temperate Lakes LTER, which is where most of this data came from, the Wisconsin Department of Natural Resources, and the Ojibwe land upon which this research has taken place, as we are nested within the ceded territories. Hopefully I left time for questions.
Yeah, thanks so much. That was a great presentation and really cool. I didn't realize, I forgot in 2012 and 2013, like how drastic that must have been for station to have a fleet vehicle go through the ice and then suddenly undergrads are showing up and you have no lakes for them to get on. So with that, if anyone has any questions, you can go ahead, put them in the chat, or you can feel free to unmute and ask them directly. Can you see the chat, Gretchen, or do you want me to? I can always read them out if need be. We don't have anything yet, but
okay.
Just in case.
I see a kind comment from Mark.
Perfect.
I see one from David. It says, how feasible is it to use really good data sets from long-term research studies to predict ice on off dates for other non-monitored lakes? I think that's a good question. And I definitely think there are regional patterns. And so I think that you can interpolate regionally based on the size, depth, and shelter around the lake. So I think that there is some good feasibility. Precision, I guess, would be a question if you were happy with a plus minus, 2 to 3 day accuracy. I think that we could probably do a really good job on some of the regional lakes since we have good data probably on 20 lakes throughout this area that's kind of continuous year to year. We probably could do a decent job interpolating to some of the different size and shape lakes for, let's say, the Tri-County area. But obviously, the farther out you would interpolate that information, I think you'd want to use some caution there. So it can be pretty variable from one region to the next.
I can ask a question. You said Adriana and Ellie did it on Crystal Bog or
South Sparkling Bog?
South Sparkling Bog. Okay. Do you think it would have changed if they had done it, or the results would have been different if they'd done it on, a really deep lake like Trout, for instance?
I do. I think South Sparkling Bog was chosen because it's small and you can actually get it plowed, right?
Yeah,
so that is a logistics thing. So I think it's 3 acres or something. And so that was the goal, was to make it a manageable lake size. But bogs are always a little bit different. South Sparkling Bog's a larger one. But even the fish, there's not really a fish community interacting, and the zooplankton's mostly rotifer dominated. And the water is darker in appearance, so it has more tannins. And so I think there's a lot of things about the bog that make that kind of unique. But I still think it's pretty impressive how severely the light going through changed the primary productivity. I still feel like that pattern would be similar. But the snow, the other thing to know is that snow accumulation on larger lakes, oftentimes you don't get as much snow piled up. For example, Trout Lake, which is one of our largest over here, because it's so windswept, it can actually blow a lot of snow off of the ice and it accumulates around the edges, versus many of our smaller lakes around here, they're so sheltered and so they never get that wind sweep. So they'll get a lot of snowpack. So I think that the snow ice interaction also is variable depending on the lake itself. Those big lakes really don't get quite as much snowpack and white ice as some of our smaller lakes up here.
Nice.
Yeah.
Todd has a question. If you want to unmute, Todd, you can.
Hi, Todd.
Yeah. How much do you think that like use of degree days, like I know a lot of people use for fish growth to handle annual variability, does that capture a lot of this, or are there maybe concerns about how condensed the season is that just measuring degree days wouldn't capture?
I think degree days is capturing aspects of this. I guess one thing I wonder about with degree days is this change in thermal stratification structure that seems to be taking place. And so one day in a lake where you have a thermal squeeze in the hypolimnion, which is kind of what we call it when that even just the physical area down there gets smaller. Like is a degree day the same in a lake where you have much warmer overall temperatures as in one year versus the next? I don't know if that makes sense, but I think we're going to be capturing some of this lake on and off condition, but I don't know how much it captures the thermal structure of the lakes themselves.
Thanks.
Yeah. See, Lauren has a hand up.
Yeah, I don't recall where all your data came from, but thinking north to south in Wisconsin and maybe especially in Minnesota, how do the patterns, especially the variability in ice off, is it different north to south? The effects in the last decade or two, and then how might that relate to, the walleye or the perch that are adapting to it different parts of the states?
Yeah. So the long-term trends are relatively consistent between the south and north. And so we had that some of my first graphs were included Lake Mendota, which is down south, and then a couple lakes from up here, Minnetonka, which I think is kind of in the middle there in terms of latitude. And so that the long-term trend seems to be holding up consistently. We do see that the variability is higher in the northern regions. And so there's a couple reasons that could be. One is in the south, they're already hitting 0 on occasion, right? To where they have almost no ice days. And so the variability can't get more variable than 0. Like they're not going to be able to go past that anymore. But what we do see is that there's more extreme changes happening in the north within the last 10 years. And so that variability seems to be greater up here than it would be down on Mendota or Wingra, which are really the other data sets that I've kind of explored. There's still increasing variability, but not as much as we're getting up in the northern part of Wisconsin. That answer your question?
That does. So the other part of it is how the populations then may differ, which, I mean, you don't know, but that would be
Yeah.
the comment on that is, the selection pressures may have been different all along and changing more rapidly up north. So
Yeah.
maybe southern populations can already handle it better, or either they're more plastic or otherwise adapted.
I don't have that answer to that immediately, but I know Zach Feiner's been digging into walleye population dynamics in relation to this question more broadly throughout the state, because the Escanaba data definitely indicated that something was going on there. So he's been trying to cultivate a data set where he could look at that from north to south. And so maybe I'll have an answer sometime, or Zach could have an answer for us sometime soon. It's a good question.
All right, anyone have any last minute questions they want to ask? You can put them in the chat again, or feel free to unmute yourself. Otherwise, I know we got done a few minutes early, but that's probably works best for you too, so you can get a little break. Looks like there's no other questions. So with that, we can all thank Dr. Gretchen Gerrish up at Trout Lake Station. And if you have any other questions or anything, let me know, and I can direct you to her email and whatnot. So.
For sure.
All right.
Feel free to reach out. Thanks everybody for coming. I appreciate that.